riluzole actually had a median survival time of about anywhere from 6 to 19 months, which was a little bit more than what the clinical trial showed, because remember, real world and general population is different than the clinical trial. And there's been a study that showed that this last stage, or the stage 4 of the disease, according to the King staging, is prolonged. So there is some survival benefit in riluzole. So people have revisited it to see. And so often this is the first treatment that's offered when people are newly diagnosed with ALS. More recently in the United States, we got edaravone, also called Radicava, and that was based off of a study that looked at the ALS functional rating scale and showed a statistically significant difference in the rate of decline of points on that scale. And so based on that study and subsequent study that was done in the open label extension for supportive data, it was approved in the United States. And for those of you that are familiar with it, it's an intravenous medication, so it comes with its challenges. It is an IV dose. It requires peripheral IV access, sometimes with a PICC line, sometimes with a port catheter, and it requires pretty frequent infusions. And then I now insert this into all my talks when I talk about clinical trials, because often people come see me and they say, okay, you talked about the approved treatments, they're okay and modest at best. What else? What other drugs you got for me? And I kind of want to emphasize that there are non-drug things that actually make a difference in people's lives. We talked a little bit about it in our little breakout room. And so one of them is actually getting seen at the multidisciplinary care center, the specialized care center. When the nerves degenerate, it causes muscle weakness, and it can affect you in a whole different variety of ways, actually. And having that multidisciplinary approach does improve quality of life, and being proactive about your care actually helps to prolong survival. And so that's why many of the specialized centers globally approach care in a multidisciplinary fashion. The second is nutritional and respiratory management. This is straight out of our practice parameters. If you look, the last updated was 2009, so we are severely outdated and really need an update on our practice parameters. But what we know even back then is that nutrition and respiratory care is really critical, and it is part of our practice guidelines. Often when we talk about respiratory care, we try to incorporate monitoring of your breathing over the course of time. And when it comes time to talk about non-invasive ventilation, or sometimes people refer to it as BiPAP, there is some hesitancy to try it. Of course, it can be overwhelming when people talk about it, but it does. This is an old study from 2006, and there's been a series of studies that have been done since then to look at the benefit of non-invasive ventilation. And so when it is incorporated and tolerated, and this is especially in people who had normal or moderate mouth muscle weakness, there was an improvement in survival. And in fact, that's from 2006. More recently, there's actually the print ahead of print hasn't even become available yet, but this is a publication in that highlights the fact that treatment of ALS is not only about drugs. There's improved survival with non-invasive ventilation, and there was a randomized controlled study that actually recapitulated the data from 2006 and many others before it. But essentially, this editorial notes that it's no longer really ethical to do randomized controlled clinical trials for non-invasive ventilation. It does shows repetitive benefit in small series of studies, and so they recommend it as soon as someone becomes symptomatic or their vital capacity indicates it. And the second component is nutrition. In the healthy population, there's normal energy demand. In ALS, there is increased energy demand and weight loss. We know, even anecdotally, before we actually initiated many of the studies in the early 2000s, we knew that when people lost weight, they seemed to get weaker faster. This was an early study that was done in 2014. Early, Terry, don't laugh. 2014. And this was done with Anne-Marie Wills at MGH, and it was a multi-center study, but it was a small study looking at high-calorie diet, high-calorie, high-fat diet. So the percentage of fat in that particular feeding was controlled and much higher, and then control, which was the standard recommendation for enteral feeding or tube feeding supplements. So all of the patients had to have a feeding tube in place and have their nutrition based 100% through the tube so that you could control it and keep a diary of it. And the patients that were on the high fat, high calorie diet or the high calorie diet did the best. And there was a goal of a modest weight gain for that study. More recently, there was a multi-center study looking at high calorie fatty diet, and the graph on the left is A. That graph is of fast progressors in ALS, and again, showed similar findings that a high calorie fatty diet actually has a potential to impact survival and improve survival in ALS. And so I always talk about reviewing the FDA approved treatments in the US for your disease, and also focusing on nutritional and respiratory care, because there's evidence to support that it can impact your disease and also impact your quality of life. So clinical trials. I had another slide in here, but just for the interest of time, I wanted to just keep it to types of clinical trials. But when you talk about investigational drugs, you know, when you want an access to an experimental drug, you're asking for pre-approval access. Pre-approval access means that it's not yet FDA approved for anything. And so there's two ways you can gain pre-approval access. One is through clinical trials, and the other is through non-clinical trial mechanisms. That's expanded access, and that's right to try. So those are the mechanisms that exist in the US. I'll focus on clinical trial access, and that's the most common means by which people living with ALS access experimental therapies. Now, often people are unfamiliar with a term called open label extension. Open label extension usually means that you've completed the controlled portion of the clinical trial, and the company will provide you, or the sponsor will provide you the drug for free, and you'll know you're getting the drug, and the physician that's treating you will know that you're getting the drug. And then there are placebo-controlled trials, or historical control, or a different control arm, or open label where there's no control. I'll focus mostly on the clinical trials and clinical development. And so this should be familiar to most people, but I just call it up here because as I go through, we're going to talk about how some of the things that are developing in the clinical trial arena is shifting the paradigm or the model of how we develop drugs, depending on what the target is. And so you saw nice presentations today about the different mechanisms and the biology of ALS. Lyle did a great analogy for us. I really loved that talk, Lyle. And so the therapeutic approaches and the investigational drugs that are in clinical trials right now try to do a couple of things. So we have several clinical trials targeting specific gene mutations. We have a whole slew of drugs for sporadic ALS that target different mechanisms within the motor neuron that we think are dysfunctional. And then there are some drugs that we think can improve the nerve and muscle communication, or we have drugs that can improve the muscle response to diminished nerve input. And of course, we have cell-based therapies to protect the nerve cells or deliver protective factors. And so this is just a snapshot, I hope you can see it, of the clinical trial pipeline in the US. And so this was based off of a clinicaltrials.gov search just this past month. And as you can see, there's a lot more early phase clinical trials than late phase clinical trials, and a variety of compounds that target different targets within the motor neuron or the skeletal muscle, or actually in the next slide, you'll see there's cell-based therapies. So T regulatory cells have been discussed heavily amongst the community, and there is an ongoing autologous, which is your own T cell therapy. And then there is actually a planned allogenic T regulatory cell therapy that's planned to launch at the end of this year. Everyone is pretty familiar, I think, with the modified autologous mesenchymal stem cells from Brainstorm. And then I just wanted to direct your attention here. You know, when I first started practice, I could only really check for SOD1 on a genetic panel. And our panels have grown, and our knowledge of genetics has grown immensely. We talked a little bit about it during the introductions. And so there's an ongoing clinical trial with an antisense oligonucleotide, which is a technology to target the gene for SOD1. That same technology is being applied for C9orf72. And there is actually the same similar technology being targeted for ataxin-2. And just this past month, the same technology has been applied for a more rare condition associated with the FUS gene. So why I highlighted is because when therapy enter clinical trials, we have to identify those individuals that would be eligible. So it becomes more critical to do genetic screening. So it's impacting the way we handle our care. So when someone comes in, even if they don't have a family history, we're talking about genetic screening. And more importantly, we talk to individuals that are related to the person living with ALS and carrying a gene mutation, because it becomes important for them, as we are now moving into an era where we're going to be launching this year the first pre-symptomatic clinical trial for SOD1 using tofersen. So I just showed you a whole list of clinical trials that are available. And as an individual, and actually for C9, there likely will be multiple clinical trials that would be available, even for the genetic form. For sporadic ALS, there are multiple options. And often people will come to me and say, well, which one should I do? And so this list, I can't even take credit for. I should have put her name. One of my patients, actually, she's a project manager for Goldman Sachs. And she's like, this is just too overwhelming. There's so much information. I'm trying to sort it out, but you need to help people kind of sort out all this information. So what are things that I should consider when trying to pick a clinical trial? So the phase of clinical trial is important. Is it early in development? Do we know? Usually, if that's the case, we don't really know much about the safety. Is it late in clinical trials? So there might be some signal that they're trying to confirm and a lot of safety data. So it depends on how much risk you want to take. What's the mechanism of action? For someone who carries a gene mutation, if there's a gene-directed therapy for your specific mutation, you might lean towards that clinical trial than maybe something else. Do I have a medical contraindication? A lot of people are very excited and enthusiastic about participating in a clinical trial or gaining access to an experimental therapy, but sometimes that therapy can make whatever underlying medical conditions worse. And so we have to keep that in mind. And we talked a little bit about the side effects, when we're talking about the informed consent discussion. We don't know all the side effects. Sometimes we do a phase 1 and a phase 2, and we go to a phase 3 and we start treating people outside the US or a larger population or longer, and we start to develop or find new kind of issues that are associated with chronic dosing. But that's important when you make the decision and decide, how much risk you want to take. How is it administered? When I first started, it was typically oral administration tablets. If you come in, we give you the month supply of the oral tablet of the experimental medication. But now there are trials using oral tablets, oral liquid, subcutaneous injections, intravenous administration, spinal injections or bolus into the spinal fluid. There are direct injections into the spinal cord and even into the spinal fluid in the brain. So all of these carry risks, and people make decisions based on how it's administered. The chances of placebo. So not all clinical trials have placebo, but often they do. And you have to look carefully at what the randomization is for that clinical trial. Where you'll find it is in your informed consent form, right? You talked about informed consent. And you'll see whether it's 50-50, maybe it's a 2-to-1 randomization, 3-to-1 randomization to favor your chances for treatment, or it might just be open label where there's no placebo. How frequent are the visits? I'm in New York City. And even people who live within the 5 boroughs find it difficult to come to the center. The traffic is brutal here, just as in many other places. And so the frequency of the visits, in-person visits, are a challenge. And the last is whether there's an open label extension. And so this means that after you participate in the controlled portion of the study, is there an offering by the sponsor for you to gain access to the experimental drug where you're definitely getting drug? And so these are things that people find useful in trying to consider what clinical trial to enter into. And so many of you are familiar with the SOD. I was not an investigator for the early phase clinical trial, but I am an investigator for the phase 3 clinical trial of tofersen, which is an antisense oligonucleotide. So it's a gene-directed therapy targeting SOD1. And it was published in the New England Journal of Medicine. And this is actually clearer data from the poster that was presented at the American Academy of Neurology. But one of the you just heard all this discussion about biomarkers. One of the kind of advantages to this program was the fact that there was something that you could measure in the spinal fluid that's associated with the gene mutation, the SOD protein. And you could look at that and look at how it relates to the actual dose of the drug, and whether if you increase the dose, does it go down further? The same, once you see that, trying to correlate it with clinical outcomes. So we had a little discussion about ALS-FRS score, and we could see that at the highest dose, you had the most decline of the SOD1 protein, and you also had the slowest decline of the ALS functional rating scale. So I showed you initially the phase 1, 2, 3 model. And one of the unique things about this particular trial is that it went from phase 1 and 2, instead of stopping and starting a whole new trial, they used those patients at whichever dose was chosen, which was the 100 milligram dose, and expanded it to a third stage. So essentially their early phase study kind of fed into a late phase pivotal trial. So you jumped kind of the phase 2 portion of it. And so that allowed some expediency in getting to the answer. Because it is a difficult to enroll study, because they were looking for rapid progressing mutations, specific mutations for the study. And then what I will just add is just 2 days ago, the company did announce that they are going to offer an early access program. So this is public information at this point. And so for patients who carry the SOD gene, who have more than 2 points per month loss, can apply through the website on an individual basis to the company for treatment with tofersen if they're rapidly progressing. That's to start in July. The second part of that announcement that came out 2 days ago is that in the fall of 2021, if the study is positive and they're in the process of filing for approval with the FDA, they will then open an early access program for all SOD1 mutations. So it doesn't matter which type of mutation, doesn't matter how fast you're progressing, you can then be eligible for that early access program. So that was a new announcement from the tofersen program from Biogen. The second one that's created a lot of buzz is the AMX0035. As you all know, it was published in the New England Journal of Medicine. This was a phase 2 type study. And it actually had the primary endpoint as the ALS-FRSR, and there was a statistically significant difference in the rate of decline of the ALS-FRSR. If you extrapolate it, the change from baseline is about 2.3, which is very similar to Darivone. And there was analysis of survival that showed that there was a 44% risk reduction of death. And so in that way, it prolonged survival, according to the study, about 6 months. And so it did better than riluzole in terms of that. And the adverse event profile was mainly GI, and there was fewer SAEs or serious adverse events in the treated arm versus the placebo arm. So in March, there was an announcement that based on that study result, they're going to file for a new drug submission, an NDS, in Canada. This past month, there was an announcement made by the company that in Europe, they'll be invited for conditional approval, and that they will have to do another clinical trial, mostly in Europe and some in the US, to confirm their results from this completed trial. And so I'll just add a couple of more trial highlights. So you know that levosimendan is a skeletal muscle activator. That study was negative, and its primary endpoint was actually the breathing measured laying down. And of course, Brainstorm read their data out, and their endpoint was responder analysis. So it's all kind of different takes on function. But it was looking at people who had a 1.25 point improvement in their post-treatment slope compared to their pre-treatment slope. And that's a pretty high bar, actually. But the FDA did make a public statement about their review, saying that they needed to see more data, and they didn't think it was sufficient for licensing or approval at this time. And then right around the corner, it's been kind of low on the radar, is Orphazyme. They have a product called arimoclomol. I'm not an investigator for that either. And that is also pending clinical trial results. Again, it's an orally administered drug, and it's fully enrolled. We're just waiting. Actually, people have completed, and we're waiting for the trial results to be announced. And so those are kind of what's coming down in the pipeline in terms of news. And then the readouts for these trials, and some of this has pushed us to think about what are we doing right, what are we doing wrong, and make us think about how we're doing clinical trials. So I wanted to highlight a couple of pitfalls in clinical trials that we're learning from and trying to improve and find an opportunity going forward. So we talked a lot about this already. The disease is different from person to person, and we still struggle with the biology, although we know a lot actually to target some of these mechanisms. But one of the things that from my industry experience that I've learned is that usually drugs that are entering and jumping into phase 2 for clinical trials, we really don't have a great understanding of the drug behavior or the optimal doses. And so this is often a handicap, and I think leads to late phase clinical trial failures for ALS in particular. And then also, if we're going to look, you saw how many drugs, investigational products are in the pipeline, and we only have a limited number of people. And so how can we do these more, how can we do a clinical trial more efficiently? How can we kind of conserve the time that people have to participate in clinical trials? How can we get to an answer or graduate maybe an early phase clinical trial to a more formal, larger clinical trial and conserve our resources? And then the other, we talked a little bit about this too, limited endpoints for use in clinical trials and trying to develop better ones. And then post hoc analyses, which has been a little bit of a bane of our existence. So, the approach that we've done in ALS historically has been the one size fit all, but as you know, we're slowly learning that there are subcategories of people that kind of fit better for certain types of therapies. And so that could be finding a subpopulation that might be responsive to the treatment by their demographics, by where they are in the disease, or maybe their molecular or genetic definition, or even going to precision medicine, looking at, and we're kind of doing this with the gene therapies, right? So we're looking at people who have specific gene mutations, enrolling those individuals for gene-directed therapies. So that's kind of, I think, how we're going to have to shift how we do clinical trials, trying to mitigate disease heterogeneity, the variability in the disease. I mean, one path we've taken is trying to kind of restrict the inclusion-exclusion criteria. Maybe if we refine that population, we'll unmask the treatment effect. But what we're doing is actually limiting access to investigational therapies and maybe missing an effect that might be more broader to the population. And there is FDA guidance that is pushing right now for all disease categories, not just ALS, to make a wider eligibility criteria so that whatever is being tested can be applicable to the broader population unless it's scientifically justified to narrow that population. And so trying to do stratification, you heard about the importance of biomarkers and understanding how to enrich a population, or actually looking within a population and trying to follow a biomarker, or maybe enrolling a wide set of people and then pre-specifying that subpopulation that you think might respond. This is something I've taken from my experience developing drugs from the industry side. There's often this excitement when you hear that a drug has had an effect of reversing damage in a motor neuron, or has reversed disease in a mouse model, and it gets media attention. And people get excited and they come and ask me questions about it and how can they take it. And then they ask, why is it taking so long to come to human clinical trials or for me to actually access it? And so it's really important to know that some of the studies that we do in mice, in the lab, in the preclinical setting, is really to understand and optimize the dose, right? The less we do it there, the more likely it will do nothing in the human clinical trial, or the more likely it will be very toxic. So we have to understand what the right formulation is. Does it cross the blood-brain barrier and get to where it needs to go? Is there a biomarker that we can develop along with it so that we can kind of test it in people? The safety and toxicity, of course. And then there's a stage of like having to manufacture it. So we want to make sure we have the right formulation. Can we consistently manufacture it? And will it be enough to supply a human clinical trial? So that dosing information is based on the safety and toxicity data. So I can't dose anything until I know what the range is in a phase 1 clinical trial. And so often I get asked about drugs like Procetin and NU9 that are still in the preclinical setting, but they've got a ways to go to try to get to human clinical trials. And then the last few slides are about post hoc analyses. So Eric, actually, I'm warming the crowd up for Eric's talk. And so the statistics talk hopefully will be a little bit more detailed, but I just keep it simple. I do have a master's degree in statistics, but I keep it very simple. In clinical trials, we're trying to avoid type 1 error. And this is important when we talk about reviewing articles that show clinical trial results. So type 1 error means we mistakenly conclude that there is a difference between two groups when there isn't. So we're mistakenly saying that there is a difference when there isn't. Type 2 error is when we mistakenly conclude that there is no difference when there actually is. And so we try to control for these things. And so often you'll hear a lot about the p-value, and the acceptable p-value is about 1 to 5%. So we accept a 1 to 5% chance of making a type 1 error. So meaning we're assuming that there's a 1 to 5% chance that we conclude that there is a difference when there was none. So I know that might be a little bit difficult to follow as words on a slide. So I just threw this in here just to kind of give it a little bit more flavor. So the first is the type 1 error. You're concluding that there's something there when there isn't. So the first obviously is the guy and the doctor says you're pregnant. The second is type 2 error, where you're assuming that there is nothing there, but there is, right? And how do you know when you're looking at this picture that these are wrong assumptions, right? You're looking at the context, right? So in ALS, this is one of the things that are very near and dear to me, because statistics are numbers, and it's important to avoid these types of errors when you conduct a clinical trial. So understanding the population that you want to treat, understanding how you're powering them and how you're trying to avoid those errors are important. But then when you get the trial results, the treatment effect of whatever endpoint you're looking at is important. The p-value, or the type 1 error chance, is important. And if you're significant regarding how, people will kind of look at that p-value and say, oh, it's nominally significant, it's very significant. But you got to look at the treatment effect. So you got to look at the context. And it's not just the treatment effect, it's whatever that secondary, Dr. Bedlak was talking about other measures that are measured in clinical trials. What are all of the measures look like as a whole? And then what is the safety and tolerability profile? And I think that begs the question of how we judge what is substantial level of evidence in the context of ALS. That's really important. And I think our field has done a great job of trying to develop the drug development guidance and trying to get the clinical trial guidelines published. And that was, I think, the first time before that, I think we were all in different places, and it got the community together to start this conversation. And we have a little bit more to go. And post hoc analysis, I just put it here because, if you just do multiple analyses, it's likely that you'll hit something. And when you hit that something, it's likely due to random chance. So if you, and the reason why this might happen is that drug companies, invest a lot of money in their clinical trials. And when it's negative, they're really trying to figure out or justify that investment, right? At the end of the day, I hate to say that, but that's true. And so then they have to figure out, and they'll look, and maybe they missed a signal. That's the other thing, right? So sometimes people who participate in the study or the community will say, well, there might've been a few people in there that responded, look harder, right? And that's justified. And so I'll give an example. So here's levosimendan. I was an investigator for this study. This was the phase 2 study that showed that there was an effect, a dose-dependent effect on the breathing as measured when you're laying flat on your back. And so the thing about it is that this was not the primary endpoint for the study, but it was a post hoc analysis that showed that was statistically significant. So the phase 3 was done using this endpoint, and it didn't pan out. So are all post hoc analyses bad? I'm calling back a slide from before. The Darabont study did 5 trials, and they did do post hoc analyses to identify the responders. And it was based on that post hoc analysis that defined this clinical trial population where they found a statistically significant effect. So sometimes it is important to make sure you're not missing a signal too. So it's not all bad, but you got to do it responsibly and take it into context. So everything, I hate to say this, but everything is not black and white. You got to take a step back and look at the big picture. And so I'll just kind of let people digest that. I know I'm probably stimulating conversation. The last set here is the impact of clinical trials. People tell me, oh my God, ALS clinical trials must have been on hold and research must have been on hold. But I have to say, I probably see all the physicians here more so than I ever did before COVID. And I love it. And the collaboration has been fabulous. And honestly, I've talked to more people living with the disease than I ever did before COVID. So I love that our community is driven, motivated, and connected. And that's going to give us a lot of power to move things forward. And so during COVID, we did worry about not being able to continue our clinical trials, but we kind of pivoted to do virtual visits, home draws, and home assessments. We talked a little bit about the speech assessments that people can do from home. You can do breathing from home, grip assessments from home, and you can do telemedicine visits. And there are a lot of different devices that we're now kind of developing. This is using sustained phonation, E or ah, and seeing how long you can hold it, and it correlates with one of our commonly used measures. I had to put a shout out for Dr. Bedlack, because not only does he have nice suits, he also has nice ideas. And so this is a great study that is trying to pilot a totally remote study. And so for compounds that fit that profile, not everything can be done that way, but this is a great way to go after low-hanging fruit. And the last here are some collaborations. I think Lal talked a little bit about the consortiums that are collecting samples and doing genotyping and phenotyping. And this is great. And it's really through a lot of partnerships and collaborations for funding that's allowed for this to happen. But it's been a great shift in our field to do that. So we have the GTAC study that's headed out of Columbia, Answer ALS with Johns Hopkins and Mass General, and Create Consortium out of University of Miami. And it's a multi-center. And then I'll put this here because data, clinical data, not only just samples, clinical data is really important too. We've learned a lot from looking at the placebo arms of clinical trials and a lot of what we know. So getting sponsors to put their data into an open free resource is really important because they're the ones using these the most to plan their clinical trials. So it'll be important as the community to encourage open sharing data. That's all I'm going to say about that. And then you guys are all familiar with the platform trials, but I just put this here because it's statistics to show you how a traditional development program will take longer and use more participants than using an adaptive design. So that's platform trial is a type of adaptive design. And there are assumptions. So you're assuming that there's that 10% of therapies tested would have a 30% slowing in the rate of progression. And then a whole bunch of simulations for a year and a half finally came to this conclusion. And so these are the drugs that are in the platform trial. And we heard a little bit about regimen D, which is pridopidine. But it is, I think, for people going through the process, it's a pretty exciting thing. And this is kind of what an adaptive design for platform looks like. And you'll have the handouts for the slide. I don't really want to have to belabor these. But I think in looking at future trials, I put this up here because aside from the platform trial, we're slowly shifting. I think the patient voice is being heard, and we are slowly kind of changing the way we might be doing things in the future. So I put here that maybe we could enroll a wider group of people, a wider eligibility criteria, but maybe pre-specify a subgroup for the primary analysis. So that way more people gain access to the experimental therapy, but doesn't jeopardize your assessment of treatment effect. It could be that maybe some of these people still are not eligible for the clinical trial. Is it possible for us to run a parallel expanded access or compassionate use arm? Is it possible? Sometimes drug companies don't like that. FDA is always open to it as long as they're not clinical trial eligible. Maybe we could call it an open label parallel study. That's been done in HIV. And then a more novel thought, and it's not so novel, we did it sort of with the lithium study back in the day. But as you go through the study, if you decline in disease function and you hit a certain pre-specified endpoint, so you hit maybe a 6-point loss of your ALS FRSR, before 6 months passes, you could then drop into an open label extension arm, and that could be called I call it a rescue arm. I worked with the researchers at Columbia to develop a clinical trial for the FUS program. In going back and forth with the FDA, they wanted a placebo-controlled study in a very aggressive form of ALS associated with FUS. And so I mean, all of us felt strong, the clinicians all felt strongly about not doing placebo. So the compromise was that if people declined and hit the endpoint before the end of the trial, they would drop into this rescue arm where they would be treated with the actual experimental therapy. For those that complete the trial, they would then roll over to the open label extension. And so We used a similar kind of format in the lithium study with a time to event, with the event being loss of 6 points on the ALS-FRS score. So going forward, I think, obviously we've got to think about speed, innovation, and access. Certainly the patient voice has become critically important, and data quality and evidence generation are critical items, but trying to look for other opportunities like expedited pathways or with post-approval data. People always say that ALS community can't do it, but maybe other communities couldn't, but I know we can. Innovative trial designs or using real-world evidence, that's where expanded access and compassionate use might be helpful to try to see if whatever we studied in a clinical trial population is more generalizable. So with that, I'll conclude, and maybe just, I should just leave you this. Dr. Bedleck already talked about this, the NEALS Clinical Trials Navigator. You can always call or email, depending on which thing you like. And there's a website for you to search trial. There's also a super cool resource from IMALS called the ALS Clinical Research Dashboard. That's for techie people who like moving parts. It's visually really appealing, but both contain similar data, and of course have people that can help you navigate along with your healthcare provider. So we've already talked about all this. So I'll leave it at that and take questions.
Thanks so much, Jinsy. And I'll just add that the NEALS Navigator and the IMALS Navigators are so much better than clinicaltrials.gov. The other investigators on the line I have a hard time with clinicaltrials.gov. So, it can be overwhelming. I'm absolutely sure. So if you'd like to ask a question, please unmute. We do have, it's kind of more of a comment from Greg. Gregor, are you here? You were saying that an additional consideration is that, you're excluded from other clinical trials if you're participating in a trial.
It's a gene, in any type of gene trial.
Right.
It's because for people who participate in a gene therapy, especially for things that use viral vectors, which is a one-time treatment, it precludes them because we assume that it's active in your body ongoing for your lifetime. So then it kind of gives people a challenge to enroll in other clinical trials. Sometimes for some of the gene therapies, there's a limited half-life. So some of the antisense oligonucleotides and other types of therapy using monoclonal antibodies sometimes will have limited effect in your body, but it might be longer than the standard 30 days that we use for clinical trials, but will allow you to enroll. So that is a challenge for sure.
Yeah, just thinking that, if it's an effective gene therapy, maybe you don't need any other clinical trials. A question would be, can you do a second gene therapy trial if you've done one, or is it like that's your one trial, best of luck?
That's a super challenging question because we haven't been faced with that problem yet. But, I love your forward thinking, and I don't know if anyone else has comments, but it is, I could see where if you have if you've participated in one gene therapy trial and it's a similar mechanism of action, you might not be able to participate in that subsequent study. And so that is actually something that I think companies, regulators, and the community will have to grapple with and how to handle. It's coming.
In the first SOD1 antisense trial, we did re-enroll patients in subsequent cohorts, right? So patients could have been in the first cohort, and then after that completed, and the second cohort was a higher dose, we could re-enroll those same patients, re-randomize them. That wasn't
so that was sort of a pseudo-adaptive design. So essentially, that was for them to kind of maximize those patients and bring them up to the dose that's being tested and move forward. But if you have someone who took the SOD, ASO from Biogen, and say they participated, and then there's a new therapy, actually it's not in the US yet, but there's a monoclonal antibody being developed, would they be able to enroll in that? I don't know.
I'm actually talking about the one before, the original.
Oh, the original one, yeah.
Because I enrolled those patients, right? And I snaked their spinal fluid for our biorepositories. But yeah, in that original one, it was purely, you could just but you're right. I mean, and the real truth is we don't know, right? I mean, who knows? I mean, we want to get clean data.
And what about the example? I don't know, Jinsy, what you think, but let's say talking antisense against SOD, and perhaps ataxin-2, because ataxin-2 that you mentioned, and you put it in the gene therapy, rightly so, in reality is really a gene therapy approach for the vast majority of the
for
patients.
sporadic. Yeah, you're right. And so that can definitely complicate things as it competes against other trials trying to enroll sporadic ALS patients. We're in a complicated territory, you know, and
there's going to be antibody therapies. I mean, it's going to be a much more,
yeah, and it will press us to have these conversations. And actually, this is where the perspective from the community who's living with the disease and their caregivers will really need to be valuable in understanding what kind of risks you're willing to take and what you need to kind of move things forward.
I mean, my basic 2 cents, and you know, I'm sure people are going to disagree with me, but if you want to participate in clinical trials and there's a trial that you qualify for that you were offered, take it, because we clearly don't have a clue what's going to work, right? And so anyone that tells you this trial is better than that trial or this drug's definitely going to work, forget it, man. We've been saying that stuff for, you know, ever, and we don't know. And so if you want to participate in clinical trials and a trial is offered to you and it's not, you know, it's something that you're willing to do, don't look for a better trial, take it. Trials are like, there's a movie opening at the theater, but it's only showing one time and there's only one seating. And, you know, if it's something you want to do, do it.
The only thing that I will add, I agree with you, Lyle, is if you are eligible and if it is safe for you, because
absolutely,
the consideration that we have made earlier about safety, about, you know, ethically do something in an ethical way, I think that that would be a very important consideration, I think, because if you have that approach, take whatever, because I think we are walking a very fine line.
And until we have biomarkers to stratify populations and really the kinds of signals we need to determine who would respond to a particular drug, I think this is what the lay of the land is. It's the hardest thing I have. You know, we have a few different trials going on and somebody will say to me, well, which one should I join? Well, which one do you think fits you, you know, the visit profile, the, you know, the type of drug it is, you know, it's toss a coin sometimes. It's one of the hardest things I have to answer when somebody says, which trial should I join? And, you know, other people can comment on that.
I'm glad people are asking that as opposed to the majority that don't do a trial, which just baffles me. You know, in the ALS community that I am immersed in, as I heard it mentioned, people ask about NU9 or Procetin. They're willing to take that now. Hasn't even been in an animal. I appreciate doctors prioritizing patient safety because patients have the mind that nothing a twisted clinical trial or an experimental drug could do to me is worse than what ALS is going to do to me. So it's sort of a double-edged sword.
You'd be surprised, Greg.
Yeah.
And my other comment is part of that is driven by the hype around discovery. And so things that are discovered in the lab are sometimes hyped to the point of people think they're really missing out on a treatment when it really isn't ready for any kind of prime time. And, you know, that PR
oh, sorry, Terry, go ahead.
No, Lyle, go ahead.
I mean, if you want to place bets, okay, if you are getting experimental therapy, there's one of three things that's going to happen. It's going to help you, it's going to hurt you, or it's not going to do anything, right? And if you just want to play the odds, chances are much higher, looking historically, that we are either going to make you worse or not help you than
no, Lyle, don't say that.
No, and I think times are changing, and I think that we are very optimistic for the future. There's no doubt about that. But you know, when Jinsy was talking about the metabolic stuff and how we know that, you know, the weight maintenance slows down this disease and you need to have enough caloric intake, you know, where did that come from originally? The idea came from the fact that a couple of drugs that were in trial had side effects that they caused weight loss.
Yeah.
And we said, holy shit, look at that. The patients who lost weight, their disease sped up. So those drugs that actually worked in a lab model, because guess what? In a lab model, you're titrating how much you're feeding them and you're watching all that other stuff. Those drugs sped up ALS, not because they were a bad idea necessarily, but because a side effect was that they caused weight loss. And that's what made us first realize that point. So no, I'm not trying to be pessimistic or something, but it is simply a true fact that, you know, while we are optimistic and while we think some of these things are working and we've had some recent, you know, little hints of signals of success, you know, if you want to play the odds, if you were somebody at Vegas, bet against not just ALS, but bet against rational drug design in neurology, because historically we're not very good at it.
I'll add a little pearl there. You know, the weight loss is considered so negative that if I have a drug that actually shows a modest benefit, but showed severe weight loss in an ALS population, the FDA considers it a negative study and the safety as a safety, negative safety signal towards the product.
But I agree with that, don't you? I mean, I think that maintaining your weight can slow down this disease better than any drug that I can give my patients.
Well,
So,
that and BiPAP. I mean, one of, you know, non-invasive ventilation can help support survival. And my favorite lecture to give, and it was captured by, I think, a lot of everybody's comments, is ALS is a treatable disease. And really focusing on that multidisciplinary approach and the tools that we have in our toolbox now, while we look for better drugs and better ways to look for drugs. You know, we got 2 drugs that slow the disease down. Great, we use them. But we also have other interventions, the dietary, non-invasive ventilation, multidisciplinary care, which makes a huge difference to function. You know, so it's really important to stress to folks.
Yeah. Can we give the word to Deb because she asked the question in the chat that I attempted to.
I was going to call on Deb next. Deb, do you want to go ahead and pose your question? There's been a lot of dialogue back and forth. You have to unmute. Unmute, Deb.
I actually have 3 questions. I'll just pose them all and anybody can choose to answer what they want. I just wanted to know why the 3-month wait. You know, my brother had a very fast progressing form and 3 months would have made all the difference in the world for him. The second question I have is, if you have studies that you don't have placebos, is that like a real negative with getting FDA approval? And then the last question, it really is to the doctors and the scientists and the researchers. I know back in the day, I was very active in the AIDS world, and a friend of mine ended up being his caretaker. And in my heart of hearts, I believe that if it weren't for ACT UP and Gay Men's Health Crisis Center, et cetera, we wouldn't have gotten the push where now they take a pill and the virus is undetectable. And had they discovered that combination 9 months earlier, my friend would probably still be alive, right? So as advocacy groups, is there something you can advise us to do that we're not doing or we should do more?
Maybe I can take the hard one, which is the SOD one. I'll take that one. Well, you know, actually, it's a good question, and honestly, your brother's not the only one. There's actually many people that we've all may have heard from or actually interacted with directly that has struggled with this. I for one have been involved with the person that was in the media, trying to ask for and advocate for therapy. First, it's likely that the company is saying July because I think it was accurately answered. It does require, when you ask for a single patient emergency IND, it does require an application. So first, the sponsor who has publicly said that they would make it available for those progressing rapidly. It requires the physician and the sponsor to make an application to the FDA, and there is a 30-day window. Usually they give a response sooner. After that window, and I'm going to say they sometimes do it within a week or 2. After that, then usually you require an institutional review board. And the easiest access or the fastest access would be at sites that are already participating in the clinical trial. That usually takes about a week or 2. And then subsequent to that, drug has to be shipped out and prepared. So some of the lag time, so they put that, I'm pretty sure that they put the timing of that announcement out because the time that someone would actually get dosed would be July after all of the process gets put in place. So the process actually starts now. Okay. Now, you know, in the case that's been discussed earlier with regards to the public case that was made against Biogen, you know, it's true that the trial was fully enrolled and people were actively ongoing in the study. Patients were randomized to placebo or to the treatment, but it would favor treatment. And the study wouldn't complete until June. I think the last patient, that was their explanation. And there were people on the placebo arm, well, I don't even know. There are people within the study, we're blinded, right? We don't know who's on treatment and who's not. But there were people progressing pretty rapidly within the trial and may have been actually more advanced. I know for sure, personally, I had a person that I was caring for that was more advanced than the person requesting expanded access at that time. And they did feel that it was a little bit unfair that they had to complete the study in order to get access, and they couldn't drop out and just get compassionate use if that person who didn't get a chance to enroll in the clinical trial would then get expanded access. So it was a complicated situation and bad timing. But I'm hoping that manufacturers will think about this going forward, especially if there's, you know, their publication did indicate some preliminary efficacy. So if that was the case, then, you know, sure enough, they should have anticipated that the trial would be fully enrolled. There would be newly diagnosed folks. What are they going to do with those folks in that in-between time? That could be true for any compound being developed for ALS. So that's why I talked about rescue arms, parallel studies.
I love that. Yeah, I love that.
I hope that would be the wave of the future.
So basically, just so I understand, so because there might be people who were placebo group, they couldn't have access till July, then the person not in the study at all who needed it couldn't get the medicine either.
Right. If you go to their website, you can actually read their statement where they said they wanted to prioritize the people that are participating in their clinical trials and who may be on the placebo arm or who may be on treatment arm that are progressing first. They wanted to make their priority for access to those individuals first before allowing more broad access through compassionate use or expanded access. So it's a difficult position. There's no easy answer for this, you know, but I'm kind of glad you asked the question, and I wanted to take a stab at answering it because it's a conversation our community needs to have. The second is FDA.
Can I ask a question about that before you go on?
Yeah, go ahead.
The second question.
Yeah.
So this is where, because I'm obviously advocating for the person, so thank you for fighting for her, because I'm being pretty vociferous and in support of that community of people. So the FDA EAP rules say if you qualified for trial, you can't get a EAP. So while I understand the frustration of the person in the placebo, the law prohibits her from participating in an intermediate expanded access program. So that's the FDA's response, trying to stop that situation from happening to start with, right? So that part made me go, that doesn't make sense. And the other part that I'm always troubled by is when the manufacturers keep saying, but this is going to hurt people in the future. And the FDA looked at that too, right? And I know that was part of the response where they said, oh, you know, we're worried about jeopardizing future trials. And I'm like, no, the FDA has looked at 11,000 EAPs and never once has it damaged it. So I'm troubled with that.
So this is where I think the community talked about advocating. We need to convey credibly to those stakeholders, I hate using that word, but those people, the manufacturers and the regulators, that we understand the importance of completing your participation in a clinical trial. We will not try to hurt a clinical trial by trying to game the system. We're educated, we know that, we know the importance, but we want to make sure we provide access in a responsible manner. And we as a community are educated on that and understand that. That's how we need to convey ourselves to them and push them to provide that access.
Well, especially because Biogen did the right thing with the OLE, right? So for somebody to drop out of the trial, you're foregoing your 5-year OLE. So it's like, wow, I don't understand the denial.
And the second point, you had another point, I missed it. I forget what the second point was, but
Deb or me?
No, there was something else that I wanted to say, but I can't think of it, but because I'm still stuck on Deb's question about the FDA and no placebo question. But I think it's important, just as this is where advocacy groups can help to educate that we were not going to try to hurt clinical trial development going forward. I think that's an important concept, and to do it credibly. And so that's important. It's true, FDA does require placebo. They are not really, actually, if you go to the drug development guidance, even the draft guidance, they don't like historical controls for a variety of reasons. And there's ways in early phase clinical trials to avoid placebos or use matched controls, but in later phase clinical trials, they still require it. But it's our job to figure out as a community, is there an innovative way to meet their request, but allow it to be patient-friendly. So crossover designs, randomizing in favor of treatment versus placebo, and or reducing the time of exposure to placebo, or doing that rescue arm where people, if they progress, then get access to the therapy through an open label extension. So those are the ways we have to kind of go back and forth with their requests. So,
Jinsy, a question related to advocacy. You made an analogy with an example of the AIDS field that was successful in advocating and doing that. So why? Because it was a pandemic. It was something that affected so many people in a way, I mean, or in the same the analogy of COVID, right? And therefore in the ALS field, this is still an orphan disease. I would like to understand and to help Deb answer in terms of what can advocacy group do more, being also mindful of what the FDA requires, right? Because that is important.
Yeah, the FDA, what the FDA requires, they will always, a clinical trial eligible. That's the one. Michelle, you said, how can somebody who is clinical trial eligible, but the trial is closed, then be denied expanded access? That definition can be loosely interpreted. So the definition of clinical trial eligible is imprecise in the regulations.
In the guidance document, or
no,
in the
legal
CFR? Okay.
In the legal regulations. Why I had to tease this out, right? Because people ask me questions and I was like, that doesn't even make sense. So you have to ask questions. So there isn't any added verbiage in the legislation that says, oh, someone is not eligible because they don't have access to the clinical trial because they don't live near a clinical trial site. That could technically make somebody ineligible. Or the trial is fully enrolled, they no longer have access. That makes them ineligible. So the verbiage is missing. And it probably is intentionally missing, because there might be some room for interpretation. But this is where I think the community could advocate for the change to understand if these scenarios occur, can we try to do this? Because we know we can complete. There are enough of us that want to participate in clinical trials. We can do that and prioritize it, but please try to find other ways of access so that we can not only provide access, but also provide information that could be supportive. And that approach FDA could really like. But FDA will always lay back on their regulations that govern them. And when it comes to comparing them against EMEA, like European regulators and Canadian regulators, they have different rules, laws that define substantial level of evidence for marketing authorization or approvals. So that creates kind of disorganization or incoordination of like how people review clinical trial results. So regulations for them to provide conditional approval don't exist in the U.S., right? That's one of the bills that are proposed, right? The Promising Pathways Act. So they need, in order for them to have, but they do have some pathways to kind of speed up development, right? The priority review, the breakthrough designation, orphan drug designation. But when we get to clinical trial results, we're always going to have a few hundred patients. That's, if the eligibility criteria remains the same, and they'll say do another trial. And I don't know if that's always the right answer.
If I can share something anecdotally, my cousin was one of the first people to get AZT in the 80s. He was horrifically ill. He almost killed himself. It made him so sick. But his doc in San Francisco said, there's stuff coming around the pike. Just hang on, just hang on. And when I talked to the legislators trying to get these bills passed, I talked to them about the importance of us having small wins, because my cousin survived long enough to get to the next drug and then got to the next drug. And he literally survived 35 years with full-blown AIDS, not even just HIV. He had full-blown AIDS, survived 35 years, and just passed away last spring from COVID. And so what I tell them is, we have to get to the first drug if we're ever going to get to the second and the third and the fourth in the cocktail. And with our sample size this small, we're never going to get a p-value unless we increase our sample size. And there's only two ways to do that: more trial enrollment or some kind of expanded access or open label like you're talking about, right? Like a parallel arm. So it worked in AIDS. And Anthony Fauci said, and I want to get your guys' thoughts on this, he said back then, 50% of the population in San Francisco of gay men is going to die if we don't give them what they call parallel track. And so he didn't convince, he kind of talked around the CDC and everybody else without getting their approval. And he basically took that stance. And so it seems to me, if 50% of the population is going to die and he supports a parallel track, well, doggone it, we have 100% of our population that's going to die. So how can they not support giving us an alternative way to get that 90% of our population into some use of drugs, whatever that looks like for them?
Lyle, you were going to say something?
Yeah, so there's three parallels with the HIV story that I think about a lot with ALS. So one of them is, and you mentioned like, what's the role that advocacy can play, right? So the funding explosion, right? I mean, because of the grassroots advocacy in, you know, HIV and ALS, there was just this unbelievable increase in the funding. And, you know, right now we have to do the clinical trials that get funded because the drug company decides to do them. It's really hard. There's a lot of great ideas in academia, and it is hard. You know, Jinsy was talking about these, what we call IND enabling studies, right? Manufacturing, tox, all that kind of stuff. You know, there's not enough funding to help bring some of the stuff that, if you asked us, what are we excited about? Instead, we end up doing the trials that a pharma company either has a drug that they're shopping for a disease, or they feel are de-risked, right? Because it's a genetic therapy, so they can, you know, they know who to give it to. And I'm going to talk about it tomorrow, but there are some specific ways that advocacy can help with that and are helping with that. And I think that landscape is changing. There's two other things I think about a lot with HIV. One of them, somebody just mentioned, it really is a disease where there were like a bunch of drugs that did like a little bit of something. And then it was just a magic combination that poof, look at that. We just stopped the disease. And, you know, similarly, it was drugs targeting different kind of general mechanisms. And this one slowed this step down, and this one slowed this step down. And the sum was greater than the parts. And so that gives me some hope that some of these things we're seeing in ALS may, in fact, eventually lead to something that is a breakthrough like that. The other thing is that, you know, the HIV researchers were really creative, and they did something that's actually kind of similar to something that Rick Bedlack is doing right now. So, you know, there were people that were called long-term non-converters, I think, right? This is not my area, but basically people who had HIV for like decades and never developed AIDS, right? And they looked at them and they said, what the hell is different about those people? Can we figure out what is different about the people who have HIV and don't get AIDS? And then can we figure out how to target that? You know, there's people doing research on traumatic brain injury who say, let's look at the people who have been the sparring partners and been punched in the head over and over and over and over for 50 years and never got TBI, same sort of thing, or woodpeckers. You know, in ALS, I think that Rick Bedlack is kind of doing that with these ALS reversals, right? And, or you know, can we figure out the people who come to my clinic, who, when I met them 10 years ago, they had a little thumb weakness. 10 years later, that's pretty much all they've got. And yet they clearly have ALS. Like, what's different about them? What's different about the reversal people? And that's something I think that actually HIV did. And it just, you know, I don't know if there's a signal there.
So understanding though that the HIV, like the cancer field has done it equally, right? So they understood that not all cancer was created equally. One, money, and two, it is easier to take the blood of HIV patients and go and look what makes that patient different. Right. We don't have that, and that's why.
Right.
And that's why I understand. I understand all of this, but and that's why I was talking about iPSCs, and I really see your point, Lyle. That is not the perfect. Is not the perfect.
Maybe it is perfect. I don't know.
It is as good as it gets, because we cannot, a day of diagnosis, we cannot take the spinal cord of any patient, right?
I fully agree with you, but we won't know what works till something works, is all I'll say. But I mean, but the biggest thing that advocacy can do is get its funding and funding so that we're not beholden to, oh, a company wants to try this drug in our disease because it's a fatal disease and it's easier to get it approved for our disease or whatever. Yeah. You know, there's a lot of good stuff in academia, and there's no way to fund those.
And actually, one question. I mean, tomorrow starts ALS Awareness Month, right? Yeah. And I really think that we could take all as a unified voice an opportunity to take the example of COVID in terms of funding, right? Why the vaccine was developed so quickly? Simply because they put so much money into the development. Jinsy, said it beautifully. The development process, what I find in the lab, takes 25 years to develop into a drug that we can put in a patient. They were very quick because the whole world came to put money in that. And so in terms of funding, that could be the analogy. Obviously, we cannot put, this was a pandemic, this was the whole world involved, but simply to use it as an excuse to say, listen, this is when you invest in research. Yeah. This is where really you can enable collaboration. Collaboration brings faster results, more efficiency, and therefore faster results. And I really think that as an advocacy group, that you really have an opportunity now to really do it.
Yeah, and
I just, if I may, just to be, so I have a, in addition to the funding, one of the things I wanted to ask you, again, the research medical community, how much are you all collaborating together? Because it seems to me this whole working in silos, I mean, that was not only was money with COVID, but it was also the world was coming together to work together, to share information, to collaborate. And totally, I'm outside that world. But it seems to me the more I learn, the more I don't see as much sharing and working together and collaboration as I would like to. But again, it might be just my ignorance because I'm not part of your world, at least that.
I'll tell you, first of all, it's improving dramatically. Second of all, so I have a friend who is a stroke doc who's involved with Answer ALS because he's helping them make their phone app thingy-majiggy, right? Mm-hmm. And he texts me like every week saying he cannot freaking believe how much the ALS researchers all work together and collaborate and how different it is from stroke neurology, right? And
Alzheimer.
Yeah.
Right. And
from Alzheimer's disease and
yeah,
other.
I mean, it's remarkable, but you know, we always can do better. And you know, I think that this is something we'll talk about tomorrow as well. But you know, one way that we can do better, and advocates have helped with this, is by making sure that funding mechanisms from public programs like the DoD insist on it, right? If you're getting money from us, you must deposit your data in a publicly available, you know, repository. There are funding, you know, nonprofit organizations, CZI. If you get funding from them, you have to agree to put preprints out of your articles, right? There are ways that your voice can be heard in that. I'm guessing Jamie's going to talk about this tomorrow, but one of the things that you guys always really inspire me and invigorate me about is your personal stories. And around some of these topics today, specifically related to these genetic forms of ALS and these therapies, I'm kind of stunned at how many of you have a personal connection to that is meaningful and has been moving to me. And, you know, I'm guessing it's going to be talked about tomorrow, but one of the best, most effective ways to advocate is when you can impress upon somebody your personal connection to something. So maybe something to think about tonight, you know, as you're thinking about what we're going to talk about tomorrow.
Say one other thing. I totally agree with Lyall. So the ALS community is really much more collaborative. When I started, I was the one, Terry, saying 25 years. Yes, so when I started 25 years ago, we weren't so collaborative. And then Niels started, and then other foundation, very collaborative. The Packard Center started, which was really on the basic research side, based on this collaboration. Now, Jinsy mentioned Niels, obviously, Answer ALS, and we have many. I will really urge, though, the new foundations and patients-based new foundation and advocacy group to use the same model. Because what I see, because even in advocacy and in private funding, now the IMALS is starting, Team Gleason, and unless they divide and conquer in what everybody does, the foundation that really advocates for clinical care, right, versus the foundation that advocates for technology or research. But if we are advocating for the same thing, 2 million foundations are not going to be effective. Look at the example of the diabetes.
Yes.
The diabetes has one foundation for the whole country, and it always said that. And the more people, the more power, I think. We really have to be careful also in now having you advocacy group working together rather than in isolation, as you were saying.
Yeah. I agree. That is so important. There are so many small organizations that, you know, they're started in memory of a loved one initially, and people feel territorial or whatever, for whatever, they're just different missions. But I definitely think we should all be working under one umbrella as well That's a really good point.
The wheel.
Yeah.
Exactly.
The one pearl I did get, by the way, by the HIV advocates, I did a pre-approval conference on expanded access actually in January 2020. And some of those advocates have now entered into the ALS space to advocate for ALS, but I did start with them way back then. And they told me that the shift in the attention, they were ALS ACT UP, not ALS ACT UP. See, I only have ALS on my brain. The HIV group, they were actually making public protests and trying to really raise the awareness. But it was when people realized that HIV could affect them, right? So the decision makers, right? So he told me that you really have to make sure that it's communicated by the advocacy groups, the risk, right? Everyone is at risk. This is how many people have ALS, but it would be more in prevalence. It's almost similar to multiple sclerosis, but because we have different survival and prognosis in ALS, you could get hit by, you could actually die of ALS. Your risk is higher than actually getting hit by a car while you're walking on the street. I mean, those are the facts that will make people pay attention. And you have to, there are a lot of disease groups, right? So you just got to keep making noise. At the end of the day. And that is, that's
I was just going to say, and the best people to make that noise are you guys. And one of our problems, Jinsy just hit on several of the points I always make. The other is to tell people, everyone who I'm looking at here has, who doesn't have ALS has a 1 in 350 chance of getting ALS. That's pretty staggering. One of the problems that, I don't want to say, one of the issues is that folks with ALS get the best advocates, or the people who are really going to pull on folks to help us are people living with ALS. And unfortunately, the time frame that you can advocate actively, in an active way where you're going to Congress or you're going places is sometimes limited because the disease can overwhelm you and your family. And so that limitation also creates a problem for our advocacy. But the people who are going to get the most out of advocacy are really our pals. We need you to go out there and help us advocate, because when we go and ask for something, it's not nearly, we can give those statistics we were just talking about, but it's not nearly as effective as when we say, and this is what ALS does, and have a pal who can literally talk about it in a personal way.
But with such a balance, Terry, because I think there's a lot of, given at least when I think of my brother and how fast he progressed and how, aggressive his ALS was, that's just a lot to ask. I'm not saying we shouldn't ask.
No, but that's my point.
We should keep it in perspective. It's like,
that's my exact point, Deb, is that my exact point is just that folks living with ALS can't be.
Yeah. Oh, okay.
That was my point. And so that we don't have the people who are living with the disease who are well enough to go out there and be that advocate all the time.
Right. And I would just hate that my brother would have had, it sounds selfish, but that at his most well from diagnosis, that he would be spending it on the steps of Washington, D.C. You know what I mean? I know it's needed. I'm just saying there's got to be a balance there.
Absolutely.
But we agree, but let's be creative. Think, Deb, about what the Ice Bucket Challenge did.
Mm-hmm.
People who didn't have to go to the, Congress. That was like a fire. That was incredible. It probably wouldn't happen, very often or as often as we want on this end of the society. When you talk to people outside of the ALS community, they always bring to you, well, you got the ALS challenge. Why do you still need money? Well, we still haven't solved the problem. That's why we still need the money. But that is to say, with everything that's, happening in terms of the way we are connected, we have to be creative.
Yeah.
This has been a really good conversation, but I think we have Eric here. And if we ever want to get to the party and get to those of us who haven't eaten dinner, we probably should move on, but we will be taking this up again. We have a workshop tomorrow where we can talk more about advocacy and what the most effective way to advocate. That's what most of tomorrow morning is. So, Eric, how are you feeling? You feel ready to go? Share your screen and get to it. And Jinsy, thank you for all, Jinsy, Lyle,
thanks.
Kristalina, Piera, thank you for all the comments. I think that was really great that we had such an active discussion. I think people could have gone on forever.
Yeah. Good luck, Eric.
Actually do, if you don't stop us.
No, I think it's great, and that's why a face-to-face setting is going to be, that'll be great. I think we're doing okay with Zoom, but face-to-face is going to be great. So Eric, you should be able to share your screen.
We can't hear you. Yeah.
Second.
Eric has a unique ability to make statistics interesting.
Can you hear me now? Can you hear me now?
Yeah.
Yes.
Okay. I'm not sure what happened before. I'll try again. Can you hear me now?
Yes.
Okay. Okay, thanks for your patience. And sorry to interrupt this conversation. Maybe I couldn't be heard before, but I was also applauding all the perspectives, and that's such a important set of conversations to have. How can we actually move this forward structurally? I'm going to dive back down into the minutia of how we do the research, but what supports all that is everyone's advocacy and keeping trials running, finding out ways to treat patients who can't get in trials, keeping money flowing, keeping all the support structures in place. So anyhow, I'm going to give you a little talk here on clinical trials. I work with some pharmaceutical companies. Part of the conversation was collaboration. Is actually, I think, one of the things that Niels and Merritt and Jeremy, starting Niels, one of their biggest contributions to the field was creating that collaborative environment. And all these companies I work with are just, I am super impressed with how much they actually care about finding good therapies. Yes, there's a bottom line, but I really do think that they have our interests at heart. So what's the talk about today? This is somewhat preparatory to looking at the paper tomorrow morning and trying to dig through how a clinical research paper is presented. And but the focus is hopefully a little broader than that, to try to help you be critical readers of any sort of clinical data, any data that might come across your plate. You're going to be advocates within the community and asked to give your view on whether one finding or another seems credible. And there's some background that I'm going to try to provide to give you a framework for making those judgments. It's going to come down to two things. One is, was the study design good? And the other is, could the result have just happened by chance? Within the framework of study design, there are really going to be two pieces to that. Were the groups that you're making a comparison between, were they comparable? And were the assessments, your evaluation of the outcomes from that comparison, were they unbiased? But anyhow, that's the preview. That's what I'm going to say at the end of the talk too. That's where we're going to try to get. So anyhow, here's my gratuitous puppy picture. Help you become good consumers, critical consumers, informed consumers of clinical data. Hopefully that wasn't my paper. So we're going to start off by trying to think hypothetically and use this scenario to elucidate some of the features that clinical trialists think about when they're designing trials, and that any reader of a paper would want to consider in trying to evaluate those data. But I want to feel like you could really imagine this situation. So you're feeling tired, and a friend says, oh, you know, I just was at my acupuncturist and they recommended this Chinese herbal because I was feeling tired. Why don't you try it? You try it for a week and you feel better. So that's the scenario. And then the question is, did the Chinese herbal help you? How do we make some judgment about whether that Chinese herbal that your friend recommended and it took for a week actually was beneficial? And there's 6 possible structural truths, structural scenarios, structural realities that may have led to that observation of feeling better after taking the Chinese herbal. One of them is that the Chinese herbal really was efficacious and helped you feel better. And that's the goal of all of our research, is to try to find those interventions that are actually going to improve the course of disease, slow the progression of ALS. But there are 5 other scenarios that could have also led to feeling better that wouldn't reflect an actual benefit from the Chinese herbal. And so we're going to look through those. And the point of that is to try to help you think about what are these ways that I could be misled and misinterpret a set of observations. So the one that might be most familiar is what's called a placebo effect or clinical trial effect. And so what would that be? It's that the expectancy, the hopefulness about taking something that your friend recommended as effective would actually help you. And because of that hopefulness, perhaps you slept better and you actually were better rested. So I don't want to disparage placebo effects as something that's not real. There can be real therapeutic benefits from a placebo, from that hopefulness, from that cognitive state. The difficulty is that that's very hard to maintain. It's not necessarily generalizable, so it's not easily replicated. It's you know, over the course of your disease, it's not going to modify its course. It may help symptomatically in the short term, but it's not what we're driving for when we're trying to find cures for ALS. So the first alternative scenario where we may be misled is something that alters our impressions, alters our expectancy, and results in a temporary beneficial effect. There's also the possibility that we were just making some biased assessment. We knew that we took the herbal. We wake up the next day and we think, oh, you know, I feel so much better this morning. But we're focused, if we're optimistic about this intervention, we may mislead ourselves by focusing on those times where the actual observation is concordant with our prior expectation. So our self-assessment is influenced by knowing that we were taking the Chinese herbal and then subjectively focusing on those times that are when we're feeling less tired. So that's another situation where the actual effect of the Chinese herbal may have not been any, but we still had this impression that we're feeling better because of the Chinese herbal. There's also, and more problematically, what's called confounding. And there's both positive and negative confounding. But imagine that at the same time that you took the Chinese herbal, a small cold that you had starts clearing up. And so across that week, you're taking the Chinese herbal, but you're also, unknown to you, getting better from this common cold that you had across the week. And so by the end of the week, you think, wow, that really worked. It wasn't, in fact, that the Chinese herbal worked. It just was contemporaneous with the improvement in your underlying health. It can also go the other direction. So negative confounding could obscure effect. It could be that you take the Chinese herbal, it was potentially effective for you, but at the same time you caught a cold. And so by the end of the week, you actually feel feeling lousy. The Chinese herbal was having a beneficial effect, but you never observed that because the cold that you got at the same time you started taking it obscures that. That would be negative confounding. So confounding can go either direction. It's part of the reason that it's such a conundrum from observational data, because we don't know, we have no way to know from looking at a set of data whether the absence of an effect is precluding us from seeing a real one, or whether what looks like an effect is actually not real. That's a primary motivation for how we tend to run our research. Last fifth is regression to the mean, that we all go through normal cycles of fatigue. Sometimes you feel good, sometimes you feel lousy. That, there'll be little stretches where you're feeling great, little stretches where you're feeling tired. And the only time that you're going to take a Chinese herbal for your fatigue is when you're feeling fatigued. You're not going to take the Chinese herbal when you're feeling great. So if there's any sort of periodicity to an outcome, or if you only start looking at a group of patients who meet some criterion, some threshold level of dysfunction, there's the potential that they're going to improve over time just by watching them, just because that's part of the underlying nature of that course of symptoms. And that gets the name regression to the mean, that you're regressing, you're returning towards this sort of middle ground. If you're extreme high, you're going to come to the middle. If you're low, you're going to come to the middle. So that's regression to the mean, can also lead to a suggestion of benefit when one's not there. And the last possibility is that it's just chance, that we just don't understand, that just something happened, that it's unrelated to Chinese herbal, you feel better, it's not regression to the mean, it's not confounded, it's just something that's outside of our combination of understanding. And that's always, excuse me, that's always a possibility. There's always going to be some noise, some natural variation in whatever system we might look at. And we'll have to evaluate whether what we do see is likely chance or not. That's another piece of the talk. So we had these 5 scenarios that were obstacles to our correct interpretation: a placebo effect, biased assessment, confounding, regression to the mean, and chance. And the top 4 are ones that we're going to address with improved clinical trial design. The last one we're going to address with statistics. So if we think about how could we improve our confidence, how could we increase our confidence that the Chinese herbal was actually effective? One way, different than just taking it for a single week and saying, oh, I felt better that week, would be if your fatigue had been long-standing, if it had been going on for a year, and as soon as you took that Chinese herbal, suddenly you start feeling better and better. So there's a very clear breakpoint concordant with that initiation of the Chinese herbal. That would give you a little more confidence than just, I'm feeling tired today, I take it, a week later, I'm feeling a little better. It would also increase our confidence further if it were reversible. If you stop taking the Chinese herbal and you start feeling lousy again, and you start taking it and you start feeling better, if you went through several cycles of that, that would suggest that that Chinese herbal is having a real beneficial effect for you. We don't yet know whether it's generalizable, but the reversibility that's consistent with your taking or not taking Chinese herbal would substantially increase our confidence that it had a real effect. The next level of sort of this hierarchy of confidence building would be generalizability. If lots of people taking the Chinese herbal all report that they're feeling better, that would suggest that there's some real effect there, that it's not just one person being sick or not being sick, having a cold at the same time. Next level would be if we had some control group. If we could compare the people who are taking with people who are not taking the Chinese herbal, and only the people who are taking the Chinese herbal will start feeling better, then that's going to increase our confidence still further. And ideally, we would randomize that and control it and blind it. And the people, we would take a Chinese herbal, we'd take a placebo pill. Some people would get the true drug, some people would get the placebo. If only those people who were on the active Chinese herbal started feeling better, and those who were taking the placebo didn't, then we would have addressed almost all of our concerns. And if we have a very large sample and making that comparison, we'll end up addressing our concerns about chance effects. So a randomized controlled blinded trial is our current gold standard within the field, and that's the standard because it addresses these concerns about bias assessment. If there's blinding, then there's no opportunity either for the participant or the clinician treating them or evaluating them to have any subconscious bias towards assuming that they might be getting better, might getting worse. If we randomize so that the only difference between people on average is that they randomly were assigned to one treatment group or another, then that overcomes or minimizes the effect of confounding, because an equal number of people who are just getting sick or an equal number of people who are just getting over having a cold are likely to be in both of the treatment groups. It also helps us separate out these effects of placebo effects and regression to the mean. And the last thing that good design leaves for us is to evaluate whether chance might be a reasonable explanation. So how do we decide about chance? When we, in the original hypothetical, we just have one person, yourself, you're treated, you feel better. It's hard to feel that that was sufficient evidence to fully evaluate the Chinese herbal. That reversibility would improve, we talked about it a moment ago, would improve your confidence, but it's still just one person. If two people took the Chinese herbal and two took the placebo, and only the people on the Chinese herbal got better, that would obviously be greater support. It's not sure if we're enough support. If 20 people took Chinese herbal and 20 placebo and only the people on the Chinese herbal got better, at that point, we start to feel pretty confident. Somewhere in the middle is where our confidence shifts from saying, oh, maybe, to, yeah, we're really pretty sure. And we need some numbers to try to help us make some judgment about where should that threshold be. So this is just for the visual learners like myself, what are we talking about? We've got, this is ourselves, our one person. We started pre-treatment, we got post-treatment, we're going to take the Chinese herbal, and on a fatigue scale, our fatigue goes down. And we're going to compare ourselves to someone who's not taking the placebo, or not taking Chinese herbal, who's on a placebo, and they don't get better. This is 2 versus 2, 20 versus 20. And so, even just visually, you can see, well, if all 20 folks are getting better and none of the 20 folks on placebo, that's a really substantial separation. We're starting to feel pretty confident. And this is the only slide with numbers on it, but statisticians love coins. So it's a situation like we've got a nickel and a penny, or a whole bag of nickels, a whole bag of pennies. We're going to flip each one, see which comes up heads. And if it's the nickels that come up heads, then we're going to trust that the nickels are some real effect, and the pennies, being the placebos, are the non-effect. How do we separate out the likelihood? So if we flipped a nickel and it came up heads, and we flipped a penny and it came up tails, so there's a benefit from the scenario, there's a benefit from the nickel, the active Chinese herbal, lack of benefit from the placebo, coming up tails. That's going to happen it comes up heads half the time, comes up tails half the time. So the combination of heads and tails, that comes up a quarter of the time. With 2 heads and 2 tails, that comes up only 1/16 of the time. With 20 heads and 20 tails, that comes up only one, whatever that is, that's beyond a trillion, 10 trillion, 1/10 trillionth of a time. That's highly improbable. To take 20 fair nickels and 20 fair pennies and flip them and have all the nickels come up heads and all the pennies come up tails, that's highly unlikely to happen just by chance. That's way outside the realm. So where is the threshold? The threshold that we usually use is what we just, a shorthand call a p-value less than 0.05. But I want to help explain what is a p-value and why do we use this as a framework for making some judgment about whether chance might be an explanation. It comes from hypothesis testing. So hypothesis testing, we dichotomize the world. We say the world is divided into 2 possibilities. Either the Chinese herbal is beneficial or the Chinese herbal does nothing. And the null hypothesis is that the Chinese herbal is doing nothing. It's no different than placebo. And the alternative is that the Chinese herbal is beneficial. That's the one that we have an interest in evaluating. We want to decide whether the data in hand supports the hypothesis that the Chinese herbal was actually beneficial. So we have two possibilities. We divide the world in 2. So we have 2 possibilities. We can either accept the null hypothesis that the observed data might have just occurred by chance, or reject the null hypothesis that the data don't support that it was just by chance, and that the alternative is perhaps a better explanation. So there's a simple single number line here. It's the probability that we see our set of results or something more extreme, given that the null hypothesis are true. And this null hypothesis, again, is that there's no effect. So if there's no effect, if the truth is hypothetically that there's no effect, and the probability that we see our results is pretty high, so that flip a nickel once it comes up heads, flip a penny once it comes up tails, the probability of that is 25%. So there's at least a 25% probability that we would have seen that just by chance. So we'd say that's pretty high likelihood just by chance. And so we're not going to accept that the nickels are more likely to come up heads and the pennies more likely to come up tails. P-value is exactly this statement of what's the probability that under this null hypothesis of no benefit from the Chinese herbal, that we would have seen some set of data. We're going to reject the null hypothesis if that probability is low. If there's very low probability that we would see our results just by chance, then we're going to reject the null and say we have support for our alternative hypothesis that the Chinese herbal was beneficial. So this is a form of proof by contradiction. We say, for those of you who remember your Boolean algebra from 8th grade math and algebra, if P implies not Q, then Q implies not P. So that might seem very obtuse. So I have a little video. This is going to be the highlight of the whole talk today, but to try to reinforce this notion of proof by contradiction, how we do our p-value testing, inferential testing. Let's see. And tell me if you can do that.
Guten Tag.
Good day.
My name is Professor Guy Gadbois, medieval castle authority from Marseille. Tell me, do you have a ring?
I do not know what a rum is.
Zimmer.
Ah, a rum.
That is what I have been saying, you idiot. Rim. Zimmer.
Okay, so here comes the statistics. Watch closely.
Does your dog bite?
No.
Nice dog. I thought you said your dog did not bite.
That is not my dog.
Oops.
Guy Gadbois.
Sorry, how do I start off? Okay. I can't see if anyone's laughing, but I hope that you enjoyed that. I love the link path through. So why did I share that with you? So he asks, does your dog bite? The clerk says, no, my dog does not bite. The dog bites him. So if that is my dog implies that it does not bite, then when the dog bites Clouseau, that implies that's not my dog. The clerk is just completing, making it transparent what's just transpired. So that's proof by contradiction. If the dog bites you, it's not the clerk's dog. If the alternative, is false, implies that the null is true, then showing that the null is false, the dog bites, implies that the alternative is true. So we don't have truth and falsity, we have probabilities, but it's unlikely that if you get bitten that it was the clerk's dog. Okay, I hope that will stay in your memory. If nothing else from the talk stays in your memory, maybe that little clip will. And you can say, why did he show me about Clouseau and the dog? And you can go back to the slides and maybe it'll make sense. There's always more to say about something. So significance, this p less than 0.5, doesn't mean that the alternative is true. It means that our results are inconsistent with the null hypothesis being true. That it's unlikely that we wouldn't have expected to see these results if there is in fact no benefit. But there's still the possibility that it's just an outlier. This threshold is just 1 in 20. So 1 in 20 times we expect something to happen that has a probability of only 1 in 20 times, it's not, we never have the benefit of being omniscient and knowing what the real truth is. We only have some data that suggests that where we should next look. The converse is true as well, that a result that's not significant doesn't mean that there's no effect there. Doesn't mean that that the Chinese herbal had no effect. It just means that we didn't have enough data, or the data was too noisy to resolve some potential effect. So, again, it's a situation where we're not omniscient, where lack of statistical evidence of benefit doesn't mean that there truly was no benefit there. it certainly means that we would need more data to try to evaluate whether there was a benefit. And it may be that in a research program that we decide there's greater opportunity, greater potential for benefit in some other direction, and we end up abandoning a course, but we never really know, unfortunately. So when we get to the paper tomorrow, there are going to be a few things to look for. One, is there some potential of confounding between the participants' characteristics and which treatment group they actually were assigned to? Were there placebo effects or regression to the mean? Were those well controlled for? Was the assessment that the investigators made of the participants blinded to their treatment? Or was there potential that their knowledge of which treatment was given might have influenced their judgment? Were the participants themselves followed over the same course of time? Was the evaluation made of the participants equivalent? And then lastly, could those results just have happened by chance? What's the possibility that what we're observing is just a chance outcome? So those are, again, the features of good study design and an evaluation of the role of chance. One last thing, if I have just another minute, because it's another cartoon. Sorry, I'm going too fast here. Other things to think about as you're trying to make some evaluation of a set of data is how many comparisons were made. If I go and I run a study and I measure your change in weight, and I measure your change in strength, and I measure your change in ALSFR score, and I measure your change in appetite, and I measure your change in how many steps you took, and I measure your change in how long it took you to finish lunch, and whether you ate 2 sandwiches on average or 1.5 sandwiches on average. You know, if I measure 500 different things in a sample of 20 people, and I've randomized them, 10 of them to get one treatment and 10 to get another treatment, and I've got these 500 different measurements, there's one of them that I'm going to find which always is better in the group that I gave the treatment A, and always is worse in treatment B. If you measure enough things, if you massage the data or collect enough disparate data, 1 in 20 times we expect a given set of observations to meet our threshold of 1 in 20 times happening by chance. So there's a concern if you either if a study looks at a whole lot of measures or applies a whole lot of models or changes what they choose to look at afterwards, or if they fail to tell you in the paper how many things they might have looked at. And so again, here's this little cartoon to try to drive this home. So this is multiple comparisons problems. And if you don't know, XKCD is kind of a fun sciencey comic, but she ran I don't know if she has a name, but woman runs in, jelly beans cause cancer. Quick, scientists investigate, and the scientists report, no, we found no link between jelly beans and acne. But maybe the woman says, maybe it's the color that causes one particular color of jelly bean that causes cancer. And so the scientists go back and they start studying. The link between purple jelly beans and acne is not significant. There's no link between brown jelly beans. There's no link between pink jelly beans. There's no link between blue jelly beans. There's no link between teal jelly beans, salmon, red, turquoise, magenta, yellow, gray, tan, cyan, but green. We found a link between green jelly beans and acne, P less than 0.05. No link with mauve jelly beans, beige jelly beans, lilac jelly beans, black jelly beans, peach jelly beans, orange jelly beans, but there was with green. And so what comes out in the news? Green jelly beans linked to acne. That does happen. You will find plenty of evidence, plenty of situations in the public media where there'll be a touted claim of one result cherry-picked out of a whole basket of different colored jelly beans. And there's, there'll be a p-value associated with it, but it won't be the right p-value. It won't be the one that recognizes the totality of the evidence that's being evaluated. So be careful of green jelly bean associations. There are a few other things, that you could look at many subgroups, different ways. I guess I'm just trying to itemize different ways that there could be multiplicity in Could be different rates of loss to follow. There could be conflicts of interest. There are other things besides sort of the top liners that I laid out of good study design and chance, but there are other things that you'd want to make a note of in evaluating some data. So, but the conclusion is, think about good study design, be aware of the scenarios that could mislead you, especially in trials that are not randomized and blinded. Remember that a p-value tells you something about chance. It doesn't necessarily tell you about the truth. It tells you about the probability that you might have observed those data by chance. And in general, be informed and critical consumers of the medical literature. And oh, I apologize. I didn't warn you, but there's a test. There's a test at the end. Is everyone ready for a test? Okay, here's some references. So what does p less than 0.05 mean? No one, well, you're allowed to check your notes if you have your notes in front of you. But, does it mean, excellent, start prescribing? Do I have any hands? No, I don't see any hands. Does it mean, who knows? It's all best accurate to begin with. Who knows what p less than 0.05 means? I just explained it. It's this counterfactual, conflict. Anyhow, who knows what it means? It means there's less than a 5% chance that these are result, or better if the null hypothesis are true. That sounds like the right answer, but it's actually, it depends on how many tests you conducted. Okay, so if you answered C, good answer, but it was, I didn't give you the full panel. You feel better informed? Hopefully, yes. Do you enjoy statistics? More than before, maybe. So, the low bar, it doesn't have to be very high. Yes, okay. That's it. That's all I got for you tonight.
Thank you so much, Eric. Now, Eric, let's give him a hand, because nobody can make statistics as interesting as Eric can. And it's fun.
And Clouseau.
And Clouseau. That's right. So, thank you so much, Eric, because I think that really helps.
Yeah, sure.
And take some of the sort of fear out of, statistics. But this is sort of a deep breath. It's the end of our academic sessions today. And now, we are going to sort of adjourn to a social gathering. And we're going to have Jamie is going to be our MC for the social gathering. And I encourage you, look, if you got to leave, you're tired, great, got it. But for those folks who want to stay, we'll have a little fun and certainly give folks additional time to talk to each other and get to know each other. We appreciate how much attention you've given so far. It's been a long afternoon, but you guys have hung in there, and that's great. Jamie, would you like to take it over?
Sure, Doctor. First of all, thank you to all of our speakers today. You guys have been amazing, and it's been absolutely wonderful. And I have been messaging some of the attendees, and they are really grateful for everything that you guys are doing. It's so motivating to them to know that there are so many dedicated people out there that are willing to fight for them and advocate for them and to learn about them. And we are truly grateful for you all. And, more to come tomorrow. But as far as today goes, great first day. We crossed our fingers and hoping that this online version would work well. And I think it has so far. And, I'm learning. I think we all are. And so we're grateful for you all. So once again, thank you.
And I want to personally thank my friends who have come and helped me out by participating. Eric, Jinsy, Bristoline, I think, has had to leave. Lyle, of course, Piera. It means a lot to me that I can call my friends to come and help educate PALS. And they're as committed. Every one of these guys, by the way, for the PALS on this call is so committed to making a difference to you, to ALS and to moving things forward. So I want to personally thank you all for helping create this wonderful atmosphere.
Yeah.
Go ahead, Jamie. Now you can tell jokes.
Oh, you want me to tell a joke, Doctor? Okay. Hey, Lyle, what's the opposite of irony?
I don't know. What's the opposite of irony?
Wrinkly.
I listen to this all the time.
And you invited him to the
I have to log off. I just want to thank everybody. This was amazing today. It really was. I cannot thank you enough. I am so glad to be participating. You guys really rock. Thank you so much.
Deb, before you leave, I just want to share something because Deb and I both work with the patient community a lot. And for those of you.
Oh, we lost you, Michelle. Or I lost
you.
All the time. Deb and I hear a lot of the good, bad, and ugly about the neurologist in the community. And I just want to tell you guys that it brings me to tears when I talk to people to have doctors like you, that the patients come to us and tell us, you've got to send more people to these particular doctors. And all of you on the call today have been among the people that we've heard repeatedly. And so I know it's hard sometimes, your practice and delivering the bad news and dealing with all of these studies, but know that the patients, when they're talking just amongst themselves, truly honor all that you guys bring to them and all that you do to make a difference in their lives. So.
And you clearly make a difference emotionally. There's a lot of ways that this news is delivered, as you know, and a lot of different ways to give care and show support. And words are so powerful. And I just can't echo what Michelle, I mean, I talk to people, as you do, every day who have ALS, and so thank you. Today was amazing. And thank you for all that you all do. I'll see you tomorrow.
Take care, Deb.
Dr. Andrews, what do cows order from?
What do cows order from? I don't know.
Catalogs.
Oh, that's a good one.
I thought it was going to be Muyus or something. Yeah.
Oh, there you go. I like that. So I know I could see Sarah Feldman and Sarah Addicks and Terry, they're all just shaking their head because this is what, and Gary, this is what we do all day long, you know.